Thank you. So I'm here today to talk to you about a project named Underworlds. And what Underworlds indexes is the bacteria, the chemicals, and the viruses in wastewater streams. And our approach is to do this neighborhood by neighborhood in Cambridge. And what we're looking for is contrast. Contrast in those indexes I just named across the metropolitan area. And what we're looking to do is find out where we can learn from healthy communities and how we can identify at-risk communities to then sort of carry our resources there. All right, so this project involves Cambridge. We're extending into Boston in the fall, and we're also deploying in Kuwait. And the people involved in this project are several MIT labs. One is in the Urban Planning and Architecture Department. One is in the Distributed Robotics Systems Department. Another is in the Civil Engineering Department. And also in Kuwait, we also have partners in the nutrition sciences and also some public policy. So to begin, right, why sample in neighborhoods? And the first answer is we can attribute the things we measure directly to residential populations if we're measuring neighborhood by neighborhood. That makes sense. Another thing, in beginning this project, we hypothesized that the signal we measured if we migrated from the terminus of the wastewater treatment, of the wastewater system, meaning the wastewater treatment plant, if we migrated that back towards the sources, we would have a higher efficacy signal, meaning what we measured would be more human origin, and we'd find less noise and degradation due to the mechanical disruption in the sewer process itself, or the sewer itself. So was that true? So what we're looking for is bacteria, human origin bacteria, and you shed bacteria from your microbiome, from your gut. And that tells you a lot about nutrition and habits, things like that. So in human stool, we see a profile, right? These four bars represent individuals, and those three families of bacteria on the left-hand side represent three different classes of bacteria. And so we think that if we see sort of these three representative families and about these volume fractions, we can say we're looking at something like human stool. It's a good first test. So when we go to the wastewater treatment plant and we sample, we see something that looks not a lot like that first human stool sample, right? The volume fractions are completely different. So we're getting more than just human origin here. So when we sampled in neighborhoods, that more resembles what we see in human stool. So this tells us that our hypothesis is good, that we are getting a higher efficacy signal when we move our sampling closer. Great. So now I'm going to talk a little bit about how we do what we do, the what, where, when, and how. And originally what we were looking for is a virus detection platform. The idea of being able to surveil for different pathogens to detect outbreaks and then plan for them in advance. And we started with this idea. The problem is, especially in the beginning of an outbreak, the viruses are very dilute in your wastewater stream. And so when we realized this, we thought, well, if we're looking for something really dilute, and that's our gold standard, that's what we want to get to, then in the meantime, in getting there, what if we look at the things that are really abundant? All right, so then we'll get there, but for right now, let's think about chemicals and bacteria. Okay, chemicals, right? The challenge with chemicals is an indexing issue. The process for figuring out what chemicals are in your sample, which is a highly composite sample, is something called metabolomics. And so what you have to do is compare the signal you get unknown to a known sample. All right, so that's hard too, all right? So that takes us to the bacteria, the human origin bacteria, which will tell us something about the microbiomes of the people that are contributing to it. So we call that a collective microbiome. All right, so we know what we're looking for originally is this human origin bacteria. So let's go to where. So with Cambridge right now, this is where we're launching our pilot study. We know we wanted residential areas, and we wanted a minimum population size too to protect privacy. So we were able to identify some candidate locations there, and we selected one that was close to our lab facilities for just ease of transporting as a place to begin. All right, we did what we call our 24-hour study, where every hour on the hour for 24 hours, we collected a sample from the wastewater stream. Then we looked at the carbon content at each of those time points to identify when exactly we had the peak carbon in the sample. And we found that was between 10 a.m. and 1 p.m. So that's sort of the time range we wanted to be sampling. The how is our engineering part. And in the beginning, what we were doing is using a peristaltic pump, placing the pump at the top of the manhole, so right on the street level, putting a tube down to the flow itself, turning the pump on, collecting just raw wastewater, raw sewage in a bottle, and bringing that back to the lab. And that had a couple hazards. One, there was a risk of exposure that were doing it, the team members that were doing the work. So that was the primary concern there, containment of the unknowns in the wastewater. So what we did is we designed our automated sampler, which gave us some process control. And the way it works is there's two main subsystems to what we're doing. There is a motion system and a filtration system. And what we do is we design our unit. Our unit is then suspended right below the manhole lid, right below where the cars drive, where we walk, right there. When we start, what happens is the unit goes down, the motion system lowers it down to the wastewater flow, at which point the filtration system turns on and starts pumping some of the flow through a filter. The filter captures the bacteria, viruses, and chemicals based on the charge of the filter and also the size of the mesh. Then we're able to expel the water back to the flow, and when it's done sampling, the motion system then brings up the whole unit, we collect that filter, and we bring that to the biology lab for full gene sequencing. And we can kind of control a little bit better how long we're sampling. All right, so these are our two prototypes, and what are you going to name a wastewater sampling robot but Mario, the most famous plumber from the video game, and his brother Luigi? So we're iterating through this process now. We're trying to find a product that works before we do our full-scale deployment. All right, and this video will show a little bit about the process and how we And these are our team members, Pietro and Jackie and Paul. And this is right at the manhole that we use, which was right on Portland and Washington Avenue here in Cambridge, or across the river in Cambridge. And this is what it looks like in the manhole. And that's our Luigi, our wastewater sampling robot. You can see the stream. It does not actually go and submerge into the flow itself. Tubes hang off the bottom and collect, intake the flow in, which is processed by a filter, which is in that plastic tube. And this filtration process is actually something that if we were to take the raw wastewater back to the lab itself, they would have to go through the process of then filtering it in the lab. So we're saving time also. Okay? All right, so now, right, we go through the whole process of how we do it, but what did we find? Okay, we found over 300,000 bacteria and over 50,000 viruses. And that is probably a very disappointing finding, right? It's probably the least interesting thing that we could have found. But the challenge with doing something, this mass catch-all, this let's see what we find in sewage issue is, when we find things, right, it's hard to figure out what this data means. And that's why visualization is so important. It's important for us to really understand what this database means, what this database of geotagged bacteria and viruses are, but also it's important because then we can communicate that to other stakeholders. Other researchers being one of them, another one being students, policymakers, or teachers, right, a host of people. So the visualization is key for it becoming accessible. And so this first visualization that we worked on comes from that, just like one sample that we took from that 24-hour study. And so this is our bacteria. And the first thing we did was organize it into the genus and the families that we see. And this is going to be available online for other researchers to look at and try to query and figure out what are the trends, what is interesting, how do we relate this to human origin bacteria, and then how do we potentially relate that to human disease. Another step is the viruses. Again, we found a lot of viruses, but what do they mean? We think part of answering that question would be to show this to other researchers and people that are interested in the field. Another visualization is so that 24-hour study that I mentioned. Every hour on the hour, we took a sample, we looked at the bacteria, and again, made another visualization. So what's going to play through now is we'll show again the genus, the family, the different classifications of bacteria, and how each of those bacteria, the quantities we found, changed over the course of a day. So the final objective is going to be a connected system. Right now we're deploying a pilot system in one location, but eventually the plan is to have a network of them and synchronize their motion to collect data and then distribute that to important stakeholders, whether that be the residents of the community, policymakers. Okay. All right. So that's thank you very much.